All right, good afternoon, everybody. Sorry for the technical setup here. First of all, this is my first time actually presenting at a conference, so thank you to the organizers for setting this up, and thank you all for sticking around. I know I'm now standing between you and lunch, so I'll make sure to try to be brief. Make sure there's okay. So again, my name is Brandon Haigh. I'm an associate director with the cell therapy process development team at WuXi Advanced Therapies in Philadelphia. I'll be speaking today about some considerations when navigating tech transfer. My current work focuses on early stage programs, so kind of bringing in new programs that have moved into the clinical stage or are moving into the clinical stage. So I'll be focusing primarily on these points. However, some of the points that I bring up will likely be beneficial for even some of those later stage programs. I like to kind of start with some level of engagement though, of course. So just by show of hands, how many of you have been part of a tech transfer in the past? Okay, great. How many of you have been part of a completely smooth tech transfer in the past? Yeah, okay, some hands went down there. So hopefully a few details and the advice I will share here will help you in any current or upcoming or future programs. First, company slides here. So a little bit about us. WuXi AppTech is a fully integrated end-to-end CTDMO consisting of a few different teams such as chemistry, biology, testing, and ATU, or the Advanced Therapies unit. I currently work within that advanced therapy branch, helping to enable our sponsors in bringing novel therapies to market. So a lot of my slides will be talking kind of from that CDMO perspective in supporting our sponsors. WuXi's offerings support sponsors end-to-end from discovery through IND filing and through BLA into commercialization. We specialize in supporting our sponsors through production of many different types of programs, such as viral vectors, AAV, and LVV production, as well as some cell therapies, CAR Ts, tumor-infiltrating lymphocytes, and the TILs, as well as MSCs, and some other ones. This is not really an exhaustive list. All of which is alongside an integrated and standalone testing service. So again, WuXi kind of tries to be your one-stop shop in the CDMO space. As I mentioned, there are many types of programs we support, and in my opinion, one of the most exciting aspects of working for a CDMO and tech transfer overall is that wide array of programs you get to encounter on a daily basis. On the screen, you can see an example of some of the program types we manage within the scope of advanced therapies. However, given the complex and unique nature of the programs, there are also some challenges and pitfalls that need to be comprehensively managed. Today I'll discuss some of those common pitfalls, followed by some ATMP-specific challenges that may be encountered when tech transferring the diverse pool of programs in our industry today. Some of these challenges and pitfalls, maybe some of them seem kind of obvious, or you may have even heard about some of them in your own work or from even the conference this week. While tech transfer is fairly well defined by agencies, the highlighted points I want to emphasize is that it should be viewed as a collaborative program. The scope of this discussion is not limited to just tech transfer between two different organizations, like a drug sponsor to a CDMO. It can also include transfers between one lab or site to another within the same organization. Organizations can start the risk mitigation process by embracing a collaborative mindset when establishing relationship and strategy for a tech transfer program. When we think about a project, we think about what success might look like. It goes into a few of the different types of runs that must be completed in the early stage versus the later stage and how those kind of develop. But overall, in the context, teams reproducing a process or method consistently and efficiently with minimal to no errors can be a good sign a successful tech transfer has occurred. It's something you must be able to demonstrate and document. Now, I don't necessarily think there's a cookie-cutter, one-size-fits-all tech transfer process, which is why I have here that standard early-stage tech transfer process in quotes. But having a target roadmap or target process for a tech transfer can be beneficial starting point to help in alignment, strategy, risk, and timeline considerations. This is an example of what I would consider a standard tech transfer model from that incoming tech transfer, bringing that process into your partner or receiving group, to a demonstration run, verification runs, and then ultimately transfer to your production team. I like to include here a kind of a gap and a risk assessment that carries through these runs, because to me, as in the CDMO space and in the manufacturing space, assessing gaps and closing risk and addressing these two pieces throughout the duration of these early R&D campaigns really is critical. But in this case, I wouldn't focus too much on the boxes or run names, as these somewhat can be variable, but instead focuses on the campaigns. In brief, the R&D campaign is designed to gather information, test, and optimize the process, while the GMP campaign is designed to qualify, file, and manufacture that clinical product. Interestingly, there are also some opportunities where you can customize your roadmap a bit to fit a sponsor's need, as long as you recognize the risk and understand the program's need based on phase and stage of program. In a more customized process, teams can use gaps and risk assessments to define the strategy that they need for the IND filing and enable the initial runs to be utilized for expedited filing, again, in the early stages of a program. Focus should be heavy on training, which I'll mention quite a bit in my talk here, gap and risk assessing, mitigations, and generating necessary information for filing. Overall, we break tech transfer into two halves, one for transfer in from the sponsor to a development MS&T group or a process development team, another from an initial team that takes that process into a production or a routine manufacturing team, each with their own milestones like the transfer, process assessment, readiness, and execution, and different areas of focus. This is one example of how this may be completed and some specific areas of focus. The first milestone target, transfer. Incoming projects require that the new team receive all documentation and demonstrate that they fully understand and can replicate existing processes. This understanding will then inform the development of our future strategies. Consideration should be to confirm the feasibility of performing the process steps, confirm the target outcomes can be achieved, and if incoming transfer is a routine manufacturing process, then replicating and transferring the current batch records or protocols, securing your bills of materials, and supporting the required sampling plans are the focus. Process gaps must be identified and analyzed using a risk-based assessment. Any discussion of process changes for early stage products will need to consider the resulting impact on later phase manufacturing. At times, early stage process changes may be introduced to ensure ability to deliver and scale into those later phases of a program. Again, gap and risk assessment should be performed to identify this issue and outline risk mitigations for anything that may represent a challenge in terms of complying with GMP regulations. A big portion of assessing readiness involves making sure that the new team and site can source materials, has the right equipment, and that the equipment is installed, calibrated, qualified, and validated as required. The manufacturing suite for the incoming project must also be assessed for readiness. Lastly, and often the most time-consuming and involved task, is to ensure that the groups are ready as it relates to personnel in both number and skills required. Materials and labor can be the most common bottlenecks for a program. Once the project has been fully transferred with identified gaps mitigated and readiness is confirmed, the final stage is execution. If the product is in early stages, successful execution typically entails completion of those verification runs that we mentioned before, both in process development or a non-GMP environment, and at least one GMP environment run to support the product's IND submission or initial clinical batches. For a technology transfer into routine manufacturing, the goal is to demonstrate comparability, which you've heard quite a bit about at the conference so far, to the original manufacturing site and produce a final product that has similar characteristics. The team will verify all of this by demonstrating their ability to monitor the process, follow the sampling plan, and meet the same product specifications during in-process and release testing. Okay, pivoting into some of the pitfalls that we really want to highlight, we've identified 8 common pitfalls of tech transfer, which have likely been experienced by a lot of you that have taken part in a tech transfer process. Let's go through them briefly and share a few mitigations we've used. Paramount pitfall to mention here, of course, timelines. Every company is looking for the quickest timeline for their program and getting it into market. Coming to a receiving or partner group while already needing a GMP product, though, likely doesn't set your team up for much success. Engage early and understand what needs to be done by when. Otherwise, a risk-based approach is recommended in those quick turnaround time scenarios. Just make sure you understand the risk involved in eliminating activities or cutting back on your deliverables in a program. Consider staggering rather than fully eliminating your deliverables in your project plan. A good example of this for timelines and timeline considerations, we really received a contract early on and we had to make it GMP quickly. This led us to collaboratively partner with our sending group, workshop deliverables that they needed, weigh all of the risk associated with the project, stagger some realistic targets, and then ultimately hitting our timelines as we needed. I'd like to tell you there's an exact amount of time from the creation of a process to get them into GMP for every program, and assure you it's really going to be the same in every project for every sponsor that comes in. However, it can vary program to program. Some considerations for groups coming in to estimate some of your time requirements include your readiness of the suite or facility of your partner, your bills of material readiness, quality documentation like creating change controls, making those changes in your process, and overall how robust your process can be. Regarding information management, it's crucial to standardize certain pieces of information at kickoff. Items like terminology, procedures, policies, templates, and data sets can be important pieces to the puzzle. Define what information is available by your sending unit and by your partner or receiving unit, and what is needed to save that team time searching and doing rework. Ensure accessibility of information to all appropriate team members as an early win. I can't tell you how much time we've spent trying to find information, or information is stored in different areas, so working to streamline that early on is always a very helpful benefit. When it comes to training, build a strong training strategy and be opportunistic. Plan in initial timelines and throughout the program, allowing for training location flexibility like at your sponsor sites and partner sites. Enabling material flexibility is an option by leveraging R&D material inventory for training purposes, and building tools like technique videos while leveraging water and R&D runs. Sometimes people learn very visually, so creating the tools to allow them to do that is very helpful. But make sure you account for experience differences as well. Adjusting training programs to accommodate variable skill sets and experience levels has proven beneficial. A factor in training that is sometimes missed, and I think I heard it being talked about a little bit, is in considering experience levels of organizations too. So not just your personnel that you're training to perform the process, but the different organizations that are bringing in and managing the process. Factors such as what kind of training and education sending units have or CDMOs have, like academic or experienced teams versus an organization that has prior manufacturing and GMP experience, can be important. When possible, fill the gaps on those teams as needed. Understand appropriate communications for each team and group involved. Consistently important piece will always be communication. In short, be wary of ineffective meetings. Meetings that are incorrectly staffed or excessive meetings can lead to delays. Assign resources thoughtfully to enable efficient communication. And one of my favorite pieces, encourage direct SME to SME discussions. Having that middleman maybe not necessary for a program. Number 5 on my list is unclear roles. You need to understand how each organization and team member need to interact with the project and when they will need to provide support elsewhere. Establish a cross-functional and RACI as needed, and discuss collaboration opportunities across both organizations, and get a clear picture of organization handoffs and drop-offs. What's most important is that gaps are identified, recognized by both teams, and addressed. A good way I found to implement this is a breakout session during kickoff between the teams. This helped the teams understand which group was getting involved and when they were planning, what they were performing within the overall work stream. For material readiness, materials can have a wide range of potential pitfalls, such as long or unknown lead times, high cost, variable quality, and suitability. Overall, some of the most impactful mitigations we've used include establishing forward-looking inventory, or creating a strategy for some of your R&D stages, and even a more obvious one, fostering a strong supply chain team, including relationships with vendors to understand your options for alternate materials should your primary materials fall through. A large part of tech transfer is defining the testing strategy and corresponding sampling plans to avoid those testing-related delays. Define testing strategies early and collaboratively. Organizations can test their sampling plan and methods by leveraging R&D runs to refine and finalize your plans and, of course, confirm requirements. Number 8 in our list of common pitfalls is in vague agreements. MSAs and work orders really should work for the organizations that are conducting the work. However, certain agreements are often left vague, leading to unclear scope of your program, different requirements, abilities, and timing. Leverage those work orders, MSAs, and other agreements to establish and define clear expectations for the 2 or even 3 organizations involved in the program. After common pitfalls, I wanted to mention a couple specific examples of modalities. So for the tumor infiltrating lymphocytes, or TIL programs, specifically, one of the common challenges on tech transfer as it related to TIL therapy we experienced was in its benchtop nature. For example, we had a procedure coming in using a commonly used well plate that you would see in academic institutions for processing the duration of their project. While not uncommon, this can prove challenging as we move into those GMP stages of the process. Leveraging a closed system, which I love the prior presentation, talking about closed systems and automation, onboarding at the early stages, closed systems and automation can really help programs move more quickly, reduce some of those trainings necessary on it, and this is what we did for this particular program that we're thinking of here. We were able to tech transfer the program in with some closed systems much more easily and quickly. CAR T therapies have a wide range of challenges I'm sure many of you have already run into in your work with your CAR T therapies. Fresh starting materials like leukopaks often have short expirations, leading to needing expedited releases to the production teams, and short windows to process the material and receiving delays can compound those issues. When it comes to patient eligibility and availability, you can have a completely ready process, but without a patient enrolled and available for donation, this may delay execution of the project plan. Variable growth can be experienced by many therapies as it is very donor dependent. Even in healthy donors, it's somewhat variable in some of the areas that for your data and growth. However, executing comparability studies and identifying the best data-based expansion schedule and equipment will help to lower the risk. We've identified 7 challenges we encountered when we're tech transferring ATMPs, so I thought it'd be kind of interesting to talk a little bit about ATMPs specifically here. Few inputs to a cell or gene therapy are as impactful as raw materials and consumables. On the screen, you can see some bullets for considerations. From their procurement to their testing, release, use, and disposition, raw materials and consumables are a critical part of any process, and one that really needs to be closely monitored to ensure success of a program. Ultimately, these risks and others associated with raw materials can be mitigated through strong control strategies, which outline important considerations in terms of material vendors and suppliers, key attributes required for your raw materials, and how materials must be received, released, and stored. I think scalability is a fairly self-explanatory idea here, but some challenges in moving from R&D scale to GMP scale across multiple patients have been observed. Challenges to plan for here include material cost increases as your scale grows, your material availability as its scale increases in duration of the processing for personnel impact in your manufacturing suite and your production teams, and your effect on cell growth as a result of scaling and process time increases. Sometimes these pieces can be forgotten about as you go into a larger scale. It's not necessarily just one and one. It's important to understand if the time it takes to complete the process increases significantly at a larger scale or just for being executed in a GMP environment. Some challenges related to new equipment are listed here. When it comes to new technology and equipment, implementation for use in a new therapy can run into challenges such as equipment cost, reliability, function limitations, custom assemblies, and process steps. Some additional considerations are incompatibility, Part 11 suitability, availability of the systems, and agency acceptance of that new technology going into your process. Recommendation here is actually to partner with those agency contacts. Yesterday I saw a pretty good presentation from the FDA about partnering and different meetings you can set up with the agency. I was excited to hear that from a CDMO perspective because it's exactly what I would recommend here in partnering with those contacts, bringing that equipment in here. But overall, include the time needed to source, receive, qualify, validate if necessary, and train on the new or unique equipment in your project plan. Again, as these systems are donor dependent, material availability can be limited. Therapies may even require multiple collections from the same patient. It's advised to allocate what is needed for processing and assess the feasibility of secondary or recovery systems for your program that will help in protecting that starting material and ensuring patient material has lowest risk and greatest chance of use in your process. It's also advisable to have a wide range of research use only suppliers to assist you in those early stage runs and have predefined alternates for your materials. While release challenges are well known, the product cannot be terminally sterilized, which a lot of presenters have talked about so far too. But emphasizing the need for additional rigor in testing, release of raw material, as well as control of the process. Some of the challenges to highlight in this category focus on validation, availability, comparability, and shelf life of a sample or your product. Your limited shelf life is the first example I'd highlight here. Traditional sterility being 14 days, while expiration of the material or when you need it to go into your patient may be a little bit shorter than that. So it results in a lot of rapid tests. While some are in place and some regulations are in place for those rapid tests, some regulations are still evolving. Sample availability and sample matrix are other challenging areas, especially in sterility validation. Since most advanced therapies require you to use the full amount of product generated during a process, sample availability often becomes a challenge, and the resulting variability in cell concentration, media composition, and other CQAs may impact the validation's design. To address this challenge, therapy developers who can't necessarily meet the sample requirements of traditional sterility may choose to identify alternate routes that meet at least the intent of traditional sterility and validate accordingly. You should be prepared to explain why the samples you are taking for testing sufficiently represent the final product. When it comes to validation and ATMPs, unique challenges are centered around how software and systems are managed. There are complex software considerations relating to chain of custody systems that can be highly scrutinized during inspections. To help ready GMP release, teams should assess need to test equipment for full capabilities or test selected scope of features for end users, as well as developing procedural controls for those identified gaps. Lastly, ensure to communicate with end users about any gaps that exist or issues that may arise and how to mitigate risk relating to data and equipment. I won't go through this entire chart right now, but my last challenge here, comparability and change management. On the screen, I just want to show a quick diagram for considering comparability when making a change to the manufacturing process. This diagram is looking at how comparability with high variability of cellular products can be demonstrated. When donor material is not available to test a process change, surrogate material may be an option. However, you must take a risk-based and phase-appropriate approach to assess both the comparability and the suitability of using that surrogate material. Document your assessment and approach and be prepared to defend it as needed. Just remember, allocating timelines to improve process is challenging but critical. It's good practice to address potential changes to the process ahead of time. If changes are being made, do it early in the life cycle. The later you make the change, the more comparability is needed. Not that I'm saying that is a problem, as it is expected, but the earlier you make it, the easier it becomes. All right, just 6 key takeaways here to summarize it up. Adopt a proactive, phase-appropriate, risk-based approach. Understand what is needed to reach key milestones based on phase of program. Assess, document, and mitigate your risk, and trim unnecessary waste from the process. Plan for an entire scope of a program and anticipate changes. Set clear expectations by leveraging your MSAs and work orders to document needs and expectations of a program. Communicate and collaborate effectively and efficiently through all your stakeholders, sponsors, and receiving partners. Ensure your teams are working together and directly communicating, and organize information in easily accessible and shared areas. Avoid silos and tribal knowledge at all costs in your programs and within your partnership. The value of including a rigorous training plan that accommodates the changing skills needed as the project progresses cannot be stressed enough. Expect turnover and conflicting priorities for your partners. Save materials, take reference photos and videos while being flexible with your ability to train at various locations and at different times in the process. Expect changes and adjust accordingly. Plan for monitoring of skills and continuous training to reduce risk of turnover and knowledge loss. Overall, you can significantly reduce the risk and complexities of many challenges in advanced therapy by fostering and partnering with strong cross-functional and collaborative teams. Speaking of cross-functional and collaborative teams, after speaking partnering here, so as I mentioned, tech transfer is a highly collaborative process. I consulted with colleagues from all these different departments to help confirm my understanding as I developed the presentation, so I really want to give them a shout out here as it's very important. I know that was not an exhaustive list and I kind of went through it pretty quickly, but for anybody interested in a few more details that go into a little bit more comprehensively outside of 20, 25-minute presentation here, we have just recently released a new ebook, so feel free to scan the QR code and you can download the new ebook and go through some of the ideas on here as well as a whole lot more. And with that, I will conclude and answer questions.
Thank you, Brandon. We have run over a little bit, but I'm sure we can squeeze in a question if anyone had one for Brandon.
Thank you for the talk. One question. Earlier in your presentation, you mentioned development runs and one GMP-like run to get the IND. I was just wondering, is that a standard practice that you guys advocate?
For one run, you mean?
Yeah.
No. So the number of runs with, let me go back to this chart here. So I wouldn't, no, it's not presenting anymore. Thank you. This is not necessarily linear or one-to-one, in my opinion. Oftentimes, depending on how robust or how, let's say, ready a program may be to go into those later stages, into their GMP campaign, there are a lot of times where some of these runs may need to be repeated. I especially like having some of those early verification runs and training runs have duplicate numbers of runs being produced so that we can boost our training, test out different equipment, start testing out the batch record and protocols, for instance, that really allow us to kind of challenge that process and streamline where we can. When it comes to some of the later stages, some of those start getting into moving from an R&D campaign or an R&D suite into the more manufacturing and GMP suite. So again, having some repetition in there for the manufacturing team or just using the manufacturing suite and equipment can be a valuable piece. And one of the pieces I didn't mention with these runs as well is that, especially for early stage programs, as you're working to qualify some of those assays, we like to use a lot of these runs for generating some of that material so that you can then use that for qualifying or validating assays, or simply just developing new or custom assays like potency and others.
Thank you.